Welcome to Tiny Expeditions, Season 5, Episode 8, the grand finale.
It's the last one.
I'm Dr. Sarah Sharman, here to help you understand the science.
And my name is Chris Powell. I'm your co-host for this great last episode of the pop-cultured season, Tiny Expeditions. We've spent this whole season looking at how science is portrayed in some of our favorite TV shows and movies. We've covered a lot of ground.
We sure have, Chris. We looked at the representation of genetic mutations and whether it's plausible that they could cause superpowers.
You mean like the turtles?
Yes, the turtles.
We also looked at Jurassic Park and the plausibility of resurrecting dinosaurs.
And we spent 2 episodes looking at the movie Gattaca and some of the ethical ramifications behind gene editing in humans.
So I have a thought now. I feel like we're creative people, Sarah.
I would think so.
I say let's unleash the creativity.
Oh, you want to write a movie script?
Exactly.
Awesome.
So for this last one, let's don't go to a famous movie. Let's make our own famous movie. And then follow the script we did for the other episodes, and we bring in one of our researchers and we pitch it to them and see if they think this is plausible or not.
That's great. Let's go to the writer's room.
All right, excellent. So we're in the writer's room, and I would expect there to be like 10 people here, but apparently it's just you and I.
Yeah, sorry, low budget film.
That's fine. We got this. We're going to do it. We need all of the basic building blocks of a story. We need a plot line. We need characterization. We need conflict. We need resolution. Let's start with setting. Where are we going to set this?
Well, we're in Alabama right now.
I see. So let's make it familiar to us. Let's just broadly set it in the United States.
Okay.
Okay, so it's the United States, and let's just go straight for the conflict, right? It's the United States versus someone. Who's our greatest enemy right now?
Let's think this year, North Korea, aliens, anybody else?
No, you got to think bigger than that.
Okay.
Canada.
Isn't Canada really nice, non-confrontational, kind of mind their own business?
Yeah, you're talking about Canadians. Those are great people. Yeah, we've got no problem with Canadians at all, but what we're going to deal with is specifically the Canadian Maple Syrup Reserve in Quebec.
Did you just make that up?
No, I promise. It's a real thing. It's a real thing. But for our film, it's going to run out. So here is Canada facing a shortage of maple syrup.
How will they eat waffles?
That's the point. We've got a whole 2-hour movie right there just with that. I mean, you have no waffles. That's going to trickle down to the U.S. No waffle houses.
No Waffle House? Where will college students eat at 3 o'clock in the morning?
Where will I eat at 3 o'clock in the morning? It's a conundrum of epic proportion, Sarah. That's why we have to solve this. And so Canada says, we can't stand for this. We've got to have more maple syrup. And so they see that there are vast swaths of maple trees in the U.S. So here's Canada about to invade the United States to take over our maple syrup trees, or the trees that maple syrup comes from.
So we need a solution. I have a little idea.
What's that?
What if there's a spy on the Canadian lines sending the U.S. messages about their strategy?
Exactly. Okay, I like this. The spy in Canada is called Buddy.
Buddy the elf?
He's undercover.
Oh, okay.
So there's Buddy. How do we communicate with Buddy?
Text message, obviously.
Nope, can't do that. Phones out of the question. Beepers, pagers. Okay, you've watched spy movies. Those are all, yeah.
Okay, I have a crazy idea.
What's that?
It brings in a little science.
Okay.
We encode messages in DNA and put it in a carrier pigeon.
Perfect. Well, okay, so carrier pigeons. That's not all that Canadian.
Well, I mean, we could swap it out for what would you say, a Canadian goose?
Now we're talking. Yep, Canadian goose. So we have a message encrypted in DNA, but the DNA inside of the goose or strapped around its neck?
Oh yeah, straight in the goose.
In the goose. Okay, so that means that Buddy, our buddy in Canada, has to know how to sequence DNA.
Yeah, but that's a minor plot line.
Okay, so he gets these messages and he's able to foil the plan to invade the United States to take over our maple trees once he gets that message from the DNA.
Yep, Waffle House saved.
Perfect. We need to run this by our faculty investigator, but I think this is flawless.
Yeah, we're gonna make millions.
Sure, I'm Brian Roberts. I'm a researcher at HudsonAlpha Institute. My expertise is in I guess I'd say functional genomics, which is understanding how the genome actually works and how it accomplishes what it does in terms of expressing genes and making different cell types do different things and responding to different environmental stimuli. So most of my work is in manipulating cells in a dish. Usually those are derived from induced pluripotent stem cells that we turn into other cell types. Most of the time it's neurons, but we've also played around with other cell types as well. And then at least half my time, probably more, is in computational work, which is analyzing the very complex data that comes out of those cell-based experiments and trying to figure out what we did and what we might have learned, and also developing new techniques so that we can learn new things that we couldn't before.
Before we get into your work specifically and how that kind of coincides with our brilliant screenplay that we have put together, we need to ask you, though, do you have any favorite science fiction movies, TV shows, anything that sticks out to you where the science is being portrayed?
So in terms of my favorite science fiction, it would probably not be very accurate science fiction. So I'm a big fan of the original Dune, not the...
Oh.
I didn't mind the two that came out recently, but the weird David Lynch version that came out in 1984, that's one of my favorite science fiction movies. In terms of the accuracy of the science, there was a one about, it wasn't Outbreak, it was one of those pandemic movies that came out. Oh gosh, I can't remember. I think it had Kate Winslet in it.
So we've done a little bit of fact checking, and we believe that this film with Kate Winslet is Titanic.
Chris, there was not a pandemic on the Titanic.
Well, not that they recorded anyway.
The movie is actually called Contagion.
Okay, makes more sense.
The science was pretty good in that. So, one of the things, they were trying to find a cell that they could culture the virus in so they could get enough of it. And so when I saw that part, I was like, that's something that you would actually have to do in this kind of process. So I think they had a pretty good consultant for that one.
So does it ruin your movie watching experience if the science is just like wildly inaccurate, or can you find entertainment value in it anyways?
Kind of. I think so. If you're setting something far enough in the future, then I think you're in a much better space, because then as long as you're staying sort of consistent within the rules you've set up of like, we have this future science and it can do this and this and this, and you kind of stay in that framework, then me as a viewer, I'm like, okay, they're keeping in touch with their rules. But if sometimes there'll be a movie that's only set 50 years in the future, and we've got, spaceships flying at above light speed and all this kind, and it's like, I don't think we're going to be there in 50 years. That seems, and I never understand why you do that. Like, why not just set it 200 years in the future or 500 years in the future? Like, you can do whatever you want.
A premise of the movie that we were working on putting together is that data could be stored in DNA and then put inside of a pigeon, which turned into a Canadian goose.
Oh, that's right.
Yeah, we changed that to a goose. Let's just start with the first part of that premise, right? That data can be stored in DNA. Is that a legit thing?
Sure. Yeah. I mean, I don't know why. The 4 bases of DNA would be enough to encode any piece of information that you want. Now, is it an efficient way of storing it? I don't know. I mean, all digital information is stored as a bit, which is a 1 or a 0. So essentially that's a 2-letter code. So at each position in DNA, you've got more information that's storable because you have 4 different letters. And in terms of, say, the molecular size of what you're storing, DNA is going to be smaller, most likely, than a bit on a microprocessor. I think the challenge is encoding the information into the DNA and then getting it back out. And actually, the getting it back out is now fairly trivial with modern sequencing techniques. So, we can pull DNA sequence out of samples, very quickly, in hours now, where it used to take years. The encoding it into DNA is probably going to be the rate limiting step in that, because this is, it's still chemical synthesis of DNA that is fairly time consuming and slow. And so if you're talking about a very complicated message, which would probably need to be, I don't know like how complicated we're talking here, but if you're like encoding like blueprints to something, it would be thousands of bases, maybe millions of bases to do that. Could be even more. That would be very challenging to synthesize.
I mean, we'd have to have quite a bit of data because we are talking about Canada's maple syrup reserve here.
Yeah.
So it seems like a lot of data with that.
Yeah, and I know that I've read that companies have discussed like storing data in DNA, but I'm assuming that's just like native free-floating DNA, and we're talking about putting it in an organism. Is there going to be an additional layer of a problem by putting DNA into an organism?
So yeah, I was thinking about that. So I don't think you would want to put it into the genome of the organism, meaning in every cell part of its genes, right? So that's a very time-consuming process. I mean, it is done. I mean, this is how we create transgenic mice that are models of disease. In fact, there's mice that have humanized genes. So instead of having the mouse copy of the gene, they have the human copy of the gene. So that was usually done by taking a mouse embryonic stem cell and then making that change in that stem cell and then differentiating that into an early stage embryo and growing up the organism. And what you're actually hoping for in that case is that the edit appears in the gametes of the organism, because then you can just breed that animal and make more and more of that organism. But as you can probably tell, that process takes a long time, right? So now you're relying upon reproduction of the animal in order to make this line. So usually making a mouse line from scratch. I'm not a mouse biologist. I don't know for sure, but I think you're talking about a better part of a year. Maybe there's specialized labs that can turn around faster than that. So that would probably be inconvenient for sending messages. I think that what you could do is use a retrovirus. So a retrovirus like lentivirus is something that we use in the lab every day. It's derived from the HIV virus. But all retroviruses will take their genome and convert it. Their genomes are RNA. They'll convert it to DNA, and then they'll insert that into the host genome. And so that's a way of getting a chunk of DNA, because the message in the RNA is going to be preserved in the DNA, into your host cell's genome. But you get back to the problem of what cells you want to deliver it to, right? So just like it's hard to get CRISPR into cell types, and people are looking at using retroviruses to get CRISPR into cell types, you're only going to be able to get that message into a subset of the cells. So if you're trying to put it into a Canadian goose, you could probably get it into its blood cells. So if you injected it with the lentivirus, now there's a decent chance it would have an immune reaction you just gave it a virus. Now, you might be able to engineer your virus to not be as immunogenic as a normal virus would be. So there might be a way to make that work. And then, if it's in the blood cells, and then it's person on the other side could just take a blood sample and then maybe sequence it to find the sequence in there.
In our movie, which so far you haven't disproven, I'm still riding with our movie.
Sure.
You've just made some obstacles. You haven't disproven it. We just have to be ready for the maple syrup shortage about 2 years in advance to allow.
No, so the lentivirus you make in cell culture. So you can make lentivirus in a day or two.
Oh,
okay.
That's not
right
hard.
on.
Wait,
Yeah.
Side note, I've never heard of this virus before. So you're teaching me today.
It's a commonly used lab technique when you just want to knock in a gene. So when we're doing it with CRISPR, we're knocking in the CRISPR genes, which are bacterial genes. So they're not normally in human cells. So we're just knocking those in, or we're knocking something that will express a transgene. So a gene that maybe is already in there, but we want to make more of it. So we'll use lentivirus to knock it in that way. Very commonly done. Pretty easy to actually make lentivirus.
So now that we have our mechanism for getting the DNA into the goose, I think the real question now is, how is Buddy going to read it? Are we going to need AI for that?
Yeah, so I think you would want the recipient to have a sequence of DNA to look for. So the tricky thing about when it integrates into the host cell's genome, it integrates randomly. So you don't know where it's going to land. So finding it without knowing a sequence to look for would be really tough. So that actually might help with the sort of encryption part of it, is that even if the opposition knows to look in the blood cells of a goose, they wouldn't know what sequences to look for. Like you'd probably want, like I would call it like a preamble, like a chunk of DNA that says this is the start of the message, and then anything after this is the message. If you had that as a recipient, it would be trivial to find the DNA code. It'd be very easy to do. You would just you'd probably just do a whole genome sequence, and most things would align to the goose genome, and then you would just find any reads that had that preamble in it, and you could actually then assemble the message based on that. That would be pretty easy to do. I don't think you would need AI to do it. You could just do it with existing tools.
Complete side note, but do we know how complex the goose genome is?
I don't. I don't have much experience in avian genomes.
I was just curious if it was like 10 times more complex.
It's a hard thing. It's a hard thing to speculate on, because there's ones you're like, I don't know, it's probably like most other genomes. And then if I say that, then it turns out the Canadian goose genome is incredibly complex or something like that.
We'll look it up and have it as a side note.
Okay, yeah.
We did some deep fact checking. It's just Google. And we found that the goose genome is right over 1 billion bases. Comparing that to the human genome, which is 3 billion, the goose genome is significantly smaller than the human genome.
Are we talking Canadian geese here? Gray geese?
Canadian, yes.
Okay, good.
But I think also the advantage of this random integration is it would make it hard on people who are trying to decode it, because then they wouldn't know where to look, where to start. And, they're going to align to the goose genome, and most of the reads are going to align. So they're going to have to pick the ones that don't align and sift through those. The ones that don't align would not align for a lot of different reasons. So it could be just bad reads. It could be actually other viral genomes that are naturally in. So not necessarily genomes that goose was sick with and got during its lifetime, but genomes it inherited, because these viral genomes stay in your DNA and you can pass them on to your progeny. Those they would have to filter through. I think it could be done though eventually. So I think what would give away your sequence is it would be this fairly large contiguous set of exogenous sequence. And so there might be an approach where you could just start trying to assemble, like we used to do to make genomes. We used to just take small chunks of DNA and figure out which one fits with which, and then you can stretch them out and get the entire sequence. They could probably do that with those reads that don't align and be like, well, most of these assemble into these fairly small chunks of DNA, but one of them assembled into this, 10 kilobase region. That looks suspicious to me. That might be the secret message.
Interesting.
Well,
So go ahead.
I was just going to say, now we know how to go on defense also, just in case this scenario ever pops up.
Yeah.
I mean, if you wanted to fragment your message and integrate it in separate chunks, that could be that could make it harder to decode.
Okay, yeah.
Wow. So as we're thinking through the set, do we have to have a lab that these people are working in, or would it be too outlandish that they might have like a handheld DNA sequencer?
I don't think it's outlandish. There are, briefcase size sequencers right now. And there's a company named Oxford Nanopore, which makes a sequencer that can plug into your USB drive on your computer. I don't know how well it can do a whole goose genome, but you could make it work if you really wanted to. So it could be done out in the field. What you need a lab for is you have to take blood, and you'd have to process the DNA and isolate it and purify it and things like that. That you probably still need a lab for.
Okay.
Although I don't know, there's automated DNA prep machines. I don't think they would fit in your hand though. Probably fit on a table.
Okay. So that would be a scientific red flag for you if we had a handheld DNA sequencer in this video.
Just set it 100 years in the future, and then I could probably buy it.
Would that handheld device be loaded with the informatics software for interpreting, or would that have to be separate?
Sure, it probably could.
Yeah, I'm wanting like a one-stop shop. Like we extract DNA, we sequence it, we get our message out.
Basically, like a
Like a Game Boy.
a Game Boy or a what do you call it? The blood sugar meter.
Oh yeah.
Right. So you just prick, done.
I think you would need more blood than you get off of a finger stick, because
Just chop the finger off.
yeah, I don't know if the goose is gonna make it. I think you need a lot of its blood.
Oh no,
That's fine.
we can't kill geese in this movie.
Sure we can. Well, and one side note is that we're going to make the plot thicken because it turns out that this was actually an American goose impersonating a Canadian goose to get across the border.
I don't think we have time for that. This has been such fun discussion, but I think as our science consultant, we just wanted to know if you're greenlighting our film or not.
Oh, do I have that kind of power?
Yeah.
Sure.
It depends on if you greenlight it.
Sounds good to me.
Then you have that power.
Yeah.
Good.
Awesome. We'll check back with you about the whole lentivirus once we start getting
Sure.
into production.
Okay, so you greenlit it. Does that mean you also are funding?
I can give you like $10 or something.
Perfect.
All right.
Done. It's low budget.
Cool.
All right.
No, that was
Let me cut
good.
that.
Thanks so much.
I think that was a glowing endorsement from our faculty investigator. Our screenplay is greenlit. It's going to be coming to a Blockbuster store near you, the VHS aisle.
Assuming that someone wants to buy our script. So if you're interested, we're here. We could use a couple milli.
Yeah, we'll let you know where to send it. So thank you for joining us on this season of Tiny Expeditions. We hope you've enjoyed getting pop cultured with us. Stay tuned early next year for an announcement about our next season.
Tiny Expeditions is a podcast about genetics, DNA, and inheritance from the HudsonAlpha Institute for Biotechnology. We're a nonprofit research institution in Huntsville, Alabama, with a unique mission.
We bring together scientists and companies to develop and apply genomic advances to make a better world. That's everything from cancer research to agriculture for a changing climate.
If you enjoyed this episode, swing by your favorite podcast app and hit that subscribe button. While you're there, consider leaving us a review. It really helps us spread the knowledge.
Thanks for joining us.